Hello, glad you can join me today. Today I'm going to kind of explain gene mutations. This is the OCR spec. It will be 6.1.1a. Okay, so this is what you should know. For humans, there are 20 amino acids. The DNA triplets can be degenerate, which means that more than one specific triplet code can code for a particular amino acid. The DNA code is also universal, and universal will mean that it can be in any organism, on all organisms. Okay. A mutation is a random change to genetic code of an organism. Mutations may change the chemical structure of an individual gene, which produces a different allele of a gene, or structure or number of whole chromosome, and that will be a chromosome mutation. It can change the position of the genes on the chromosome. Mutations are unpredictable, they're random, they are the source of variation. Most are not beneficial. They're in a single cell, and chances can be increased by using mutagens. Mutagens include X-rays, deaminating agents, alkylating agents, base analogues, and viruses. Now, there are two types of mutations that you need to know about. You have point mutations. Now, in point mutations, it's normally a base substitution. This is just when one base will take the place of another. And there are three types of base substitutions you can get. You can get a silent mutation. Now, this is when the base that is replaced will still code for the same amino acid. That is why it is degenerate. It's a degenerate code. So if you take one base and you swap it, it can still code for the same amino acid. Then you've got a missense mutation. Now this is when you change the base and it ends up coding for a different amino acid. When this occurs, it will change the primary structure, and therefore the tertiary structure and the overall protein may not function. And finally, you've got your nonsense mutation. With a nonsense mutation, a stop codon is added. So, now most of these mutations will be harmful, but there are some that are beneficial. The ability to digest lactose is thought to have been a recent mutation. And a mutation which can be harmful is sickle cell anemia. Here, the red blood cells will develop abnormally when in low oxygen, and the gene coding for hemoglobin will contain a mutation. That would be a substitution one. Now, I said that you need to learn too, the second type of mutation you need to know is called INDEL mutation. These will affect the base pairs succeeding the site of mutation. So while with point ones, none of the other, unless you've got a nonsense mutation, none of the other base pairings or codons are affected. This one there is effect. So now the two types of indel you need to remember is in the name, indel. So if you do the in bit first, that's insertion. That's when one base pair is inserted into the DNA sequence. So all the rest of them, if you've got a sequence of like ATCG, then I suddenly input another A, it will become AATCG, and so that will change the structure. Then you've got deletion, which is the del part of indel. One base pair is removed from the DNA sequence, and so once again, if I say I've got AGTC, and I'll take away the G, now it becomes ATC. So that means that for a deletion one, all of the bases have now shifted kind of left. Alright then, now both of these changes will drastically change the structure of the protein, and so would be the most harmful mutation. Okay then, and that was me kind of explaining mutations.